I'll try and talk a little bit about biogeochemical modeling and about the BALTSEM model, and Marcus has mentioned it already in his presentation. In contrast to RCO Scooby, it's a little bit more simpler, so we model the Baltic as a chain of basins with a relatively high vertical resolution. And basically what I'm trying to do, present work of co-workers. Bo Gustafsson was supposed to give the lecture here, and BALTSEM is basically the work of Bo Gustafsson and Oleg Savchuk together. Bo Gustafsson developed the physics part, more or less, and Oleg Savchuk the biogeochemical part. And I was not quite sure what might be interesting for you and what you already know about modeling, so I mixed it a little bit. I put in some equations. I will explain how we model some processes, and we had long discussions with Maria where she put a lot of questions to me. So I'll try and answer some of those, which also show how we try and validate models. If something is completely ununderstandable, or if something is completely boring, please interrupt me. So how does BALTSEM now see the Baltic? We have already seen a slide similar. This is sort of the depth profile. If you go through the Baltic Sea, you can see that you have a series of sub-basins that are connected basically by some straits, or they are separated by some silts where the water has to go over. So what you can do is you can design a model that basically treats the area between those silts as one sub-basin and then model the water exchange between those. And that is how BALTSEM function. So basically we have 13 sub-basins, or the model divides the Baltic into 13 sub-basins. There's a very high resolution in the entrance area to the North Sea, so there are basically 7 basins till you have reached the Bornholm Basin. This is because we want to have this entrance area with a saltwater inflow in relatively high resolution. And then you take the larger sub-basins of the Baltic. This is the Bornholm Basin here. Then there's one sub-basin which is basically the Gotland Sea. So those two are the Bornholm Basin and the Gotland Sea together are the Baltic proper. So you can see that this Gotland Sea basin is actually very large. And then you have sub-basins here for the Bothnian Bay, for the Bothnian Sea and for the Bothnian Bay, and one for the Gulf of Riga, and one for the Gulf of Finland. And those basically you treat like a box with a vertical resolution, and you exchange the water between those boxes. And actually, Marcus has already shown that slide. So it is basically in BALTSEM, the physics works a little bit by the same principle. You have some parameterization of the dense saltwater inflow, and then what happens in BALTSEM is that it divides the water column in layers according to density, and if you have water coming in, then it's basically flowing down until it has reached the water layer of its own density, and then you have some kind of description what is the diffusion and the mixing between those water layers, and you also have a description of vertical advection. And what you also do, you merge water layers together if they become very similar along this mixing scheme. And in the upper part of the water column, you have a mixed surface layer, which is basically the water above the thermocline. If you work with the model, the physics basically you can't see. What you get as a model output is basically a gridded version. So you can order model output by the meter, for example, over 5 meters, but you will never see the layer structure if you don't really make it to show you its layering. So how well does this scheme actually work? Those are just some plots from our standard hindcast, and those are made for the Gotland Sea, so it's the central deep basin in the Baltic. And you can see here that the vertical resolution is sufficient to resolve things like, for example, the thermocline forming here. So this is time series from 1970 to 2009, basically. So what you see is the yellow dots here is the summer. You can see, okay, winter mixing and formation of winter water. You can see that we can resolve salinity. You can see that we get a halocline here, somewhere around 70 meters depth, and a relatively sharp salinity gradient between surface and the deep waters. And you can also see that that the model resolves the vertical gradients in nutrients. And you can see also, of course, here some parts of the seasonal cycle in these pictures. So that scheme sort of works, but it's a basin integrated scale. So if you think about our sketch, what I've shown here is sort of what we treat as an average for this entire large basin. So what we miss in our modeling approach is basically the horizontal gradients. So, but of course, for this we get an advantage because the model is computationally relatively fast, and as Marcus has already mentioned it, you can do a lot of runs, you can test parameterizations, you can do many scenarios, and you can use a lot of energy actually for calibrating, so for trying to tune it really to resolve the long-term dynamics. And it's really a model that is sort of designed to try and reproduce the long-term dynamics of nutrients and oxygen in the Baltic, because we sort of have the belief if you can reproduce the past dynamics relatively well, then you might also have a right guess about the responses in the future. So you have basically you have a time scale of response right. And then another advantage that this approach has is that you can extract information relatively easily. For example, if you're interested in a biogeochemical flux, if you want to have a rate, okay, then you just add 2 or 3 lines in your model code, you rerun it, and you have your tailor-made output. It's not that you have to think for a week whether you have really a horizontal average, and in reality your basin is not so homogeneous, and you can't resolve a small-scale feature. For example, what happens close to the coast. It's more representative for the conditions in the central part of the basin. Okay, and the physics, I think you have seen that from Marcus' slides already. Temperature and salinity, for those the model works pretty well actually. These are just some plots. This is, for example, salinity in the southern Calicut for some years, and temperature dynamics. So you can see that those are represented fairly well, and this is for, which is maybe more interesting, this is salinity in the Gotland basin, in the Gotland Sea. So you can see that, for example, here, this is things that Marcus has already shown in his slides, that it's sometimes difficult to get this deep water salinity signal right, and also to get the dynamics of the inflows right. And so, but basically the major trends are resolved in the major dynamics. So how does the biogeochemistry work then in our model? And what we know about the Baltic is, of course, that there has been long ongoing eutrophication. There has been large changes, for example, in the nutrient pools. So this is as long as we have measurements, and there has been long-term changes in anoxia and hypoxia. So this is the plot for example, for the area of laminated sediments, where you can see that there's very few laminated sediments up to about 1930, and then laminated sediments, which is a sign that there is no bottom life, so there's anoxia start to increase after something like 1940. And our aim is basically to have a biogeochemical model that sort of is able to simulate those type of long-term responses. And basically how the model works, and this is how all the three biogeochemical model works in the Baltic that Markus has presented more or less along those lines, is that you have phytoplankton, which is in BALTSEM also split into 3 groups. And those take up nutrients. You have the nutrient pools here. And then you have some kind of regeneration of nutrients in the water column to maintain primary production. And you have a variable that collects everything that is dead, and that is detritus. And the main function of detritus in a biogeochemical model is to sink and to transport nutrients basically into the sediments. And in the sediments you have pools of nitrogen and phosphorus, and you sort of simulate the biogeochemical reactions of nitrogen, or the biogeochemical processes in the reactions of nitrogen and phosphorus in the sediment, and you do that depending on oxygen condition. And in all those model processes, oxygen is always taken into account. For example, if you have autotrophs and they are primary producers, they're going to produce oxygen. If you have heterotrophic processes, for example, grazing of zooplankton on your phytoplankton, then in proportion to the grazing rate, there is going to be an oxygen consumption. The same if you mineralize detritus. This is also coupled to oxygen consumption, and also all the bending processes, they are coupled to oxygen consumption. So basically you have a model of nutrients, which is in our case nitrogen, phosphorus, and also silicate, and of oxygen dynamics in the Baltic. And I will walk you a little bit through those processes and show you how they work in our model. I will start with phytoplankton groups. I will show you a little bit how this heterotroph mineralization loop works, and I will move down to the sediments, because I think that is basically the most important thing that determines the long-term response in the Baltic. So, and I have no idea how you like mathematics, whether you like them or whether you absolutely don't like them. So this is phytoplankton, any kind of phytoplankton. And basically how we model phytoplankton, we model phytoplankton growth as a maximum growth rate that is sort of, we think, basically temperature dependent, but it's also genetically fixed. And then this growth rate is modified by the by light and nutrient. So if it's dark, you won't have any growth. If there are no nutrient available, you won't have any growth either. And if the growth conditions are good, both in light and nutrient, you will approach this maximum growth rate. And then you have a mortality term. So phytoplankton is basically dying by itself, and actually this equation goes on, so there's also grazing by zooplankton. I skipped that here. And this phytoplankton growth is coupled to nutrient uptake. So basically, proportional to the amount of biomass that is produced here, there's a nutrient uptake. So this is basically the same thing, and there's something which is the nitrogen content of the phytoplankton that you produce that links those two equations together. So this is the basic principle. Which is,
Excuse me?
Yep.
You said the growth rate is genetically fixed.
there are two things in this thing here. First of all, this maximum growth rate is temperature dependent, and then the next thing is we start with a constant, which is for the three groups of phytoplankton is different. So there's one group that grows relatively fast, and two groups that grow a little bit slower. So they say it's, for example, 2 doublings per day, and times a temperature-dependent factor, and this is the maximum growth rate that you can achieve.
When you run your entire model, is that where you start with? Like, because I was wondering anyway, you must have some initial values, like they're all connected with each other, and it's a cycle, but somehow one value you have to like use as an initiate value.
Yeah.
Is that the phytoplankton?
You have to initiate. I will come up with a list of state variables. Actually, you have to initiate each of the state variable in basically each of the depth layers that you use. Those are your initial conditions. And what we do is basically we construct the initial conditions from a long-term hindcast. And this has been run to sort of equilibrium, and then it's run from 1850 to 1970. And we use the state in 1970 as initial conditions. But basically, you have to initialize the entire system at all the physical levels in all the boxes of your model, or for macro system at all the grid points, right? Yes. So all of them. You can't start with just one. I can also show that because I have a little very simple nutrient and phytoplankton model. So basically, if you just want to model phytoplankton and couple to it dynamics of one nutrient, you have to find initial conditions for both of them, and then you can use those differential equation to model onwards. So basically, if you say that here you are at time 0, you take your phytoplankton concentration, you know your maximum growth rate, that is somewhere that's in your model constants, and you know the temperature, and you know this mortality term here, and you have to find something how to capture the effect of nutrient. But basically you can calculate this growth rate here, and then you can calculate the rate of change of your phytoplankton in this initial starting point, and then you phytoplankton is here, and then you basically simulate forward. The same you do with your nutrient concentration. So after some kind of time step, you come up with your phytoplankton concentration changes like this, and your nutrient concentration changes like this. And this is basically what you have to do for all your state variables. And what we do with phytoplankton growth, how we do this limitation, this is actually very simple. We just use this Monod type of function. We say this is the effect of nutrient is the nutrient concentration divided by some kind of half saturation constant, which we guess or take from the literature, plus the nutrient concentration itself. And this type of function looks like this. So basically, it approaches 1 if the nutrient concentrations are very high, and it's 0 if the nutrient concentrations are 0, and it's 0.5 if the nutrient concentration is the same as the half concentration, as the half saturation concentration here. So basically, for any nutrient concentration that you have, you can calculate this factor, and you can calculate your growth rate. And if we go back to my funny simple model, so what you see here, for example, when you start the model, the nutrient concentration is high, so the growth is basically exponential. Oops. And if you go forward in time, you deplete your nutrient, and then the growth rate here is basically 0. And then what you see when you continue modeling onward is that your phytoplankton biomass starts to decline again, and this is basically because this term here will be 0, and then everything is left is the mortality term here, so we have an exponential decline in phytoplankton biomass. So this is the basic principle how these equations work. And of course, what you also can do in that type of models, and this is what I've mentioned, it's very easy to extract biogeochemical fluxes from those type of models. So now we had a coupled system of phytoplankton and nutrients, and what I've plotted here is the rate of is the primary production rate at each point in time here. So basically, this is the growth term here. So you see, when you have reached there is a maximum primary production rate somewhere here, and when the nutrient concentrations decline, the primary production goes down to 0. If you want to know how much was produced in total, you basically take the integral of this primary production curve, and if you express phytoplankton and primary production, everything in nitrogen units, it's exactly the amount of nutrient that you had in the model from the beginning. So this is the basic principle if you want to put a question to your model, for example, how much carbon was produced or how much nitrogen was denitrified. This would be the basic thing that you do. You calculate the rate at each period of time, and you sum up or you integrate over, and you take the integral of this curve or you sum up during this time period. So you can also add some more realism to our model, because this is obviously rubbish. If you put something like this into a biogeochemical model, your phytoplankton will start to bloom, it will consume all nutrients, and then nothing else will be happening. So that can't be it. For example, more realistic is to say that phytoplankton is both limited by nutrients and light. And many models combine limiting effects by two factors, just by taking the minimum of each. So you can simply, similar to our nutrient limitation factor, you can derive a light limitation factor, and typically those curves have a maximum and then they decline again, because if there's too much light, your phytoplankton is going to die. If there's too little light, your phytoplankton is not going to grow. And then basically, if you have a water column, for example, where what I've marked with the lines here, with high nutrient content and low light content, you compare those two factors together, you end up somewhere here, and you end up close to 1 here. So this is a light limited situation, and your phytoplankton will grow in the model according to your light limited rate. And you can do, and for example, if you imagine it is becoming spring and finally there's more light, for the same nutrient content, hopefully, you come up now with the light limitation gone, and now the nutrient concentration limits your growth rate. And if you combine that into a model, and I've just simulated sort of a spring situation, so there's linear increase of light during the month. So just imagine this would be the difference between March and April. So in the beginning, we are in the darkness. This is phytoplankton. So there's hardly any phytoplankton growth because it's very light limited. It starts to grow. It starts to grow when the light limitation ceases. It starts to deplete the nutrients. So the limitation actually now switches from light limitation to nutrient limitation. And when it has consumed all the nutrients the entire nutrient pool, then what you see is, as we had before, the exponential decline that is due to the mortality term. So this is if you compare those two. Now, if you have both nutrient and light limitation, you can, for example, simulate a delayed growth through the spring, and this would be sort of a very simple model of a phytoplankton spring bloom. Okay, so typically, since I thought you were all biologists, what we do in our models, we have something like 1 to 3 groups of phytoplankton, which we call something like functional groups, for example, which have a little bit different properties. For example, they have different maximum growth rates. That is something from the literature. That growth rates differ seasonally, and they have usually different affinity for nutrients, so they might be very efficient for taking up, especially phosphorus. So those are groups that are adapted for growth during summer when the nutrient concentrations are low. And this is a way how you can sort of model a seasonal succession of phytoplankton groups. And then you also try to give them a little bit different properties, so some might sediment a little bit faster. This is what we usually do with diatoms. And of course, what is very important to have for the Baltic one group that is able to fix nitrogen. So these are all cyanobacteria, so this is the only way how to include this nitrogen fixation pathway into our models. There's actually something missing here. Okay. So this was a pure phytoplankton model, but basically, if you remember those slides, this is spring bloom, and then there's nothing happening again. So this is not the seasonal dynamics of the Baltic. So there's something missing here. And basically, what we need to maintain production during summer is some kind of internal recycling here. Of course, there is some kind of nutrient transport through the thermocline, also in the Baltic, and which is also captured by the physical model. But during summer, when these spring blooms are depleted, the main source of nutrients to phytoplankton that maintains the primary production is basically a recycling loop. And in our biogeochemical model, this is captured by 2 pathways, and I think all the Baltic models are pretty similar. There is one loop that goes through a detritus pool, which is just mineralized, and there's another loop that simulates sort of a zooplankton component, or heterotrophs as they're called here, and those excrete part of the nutrients they take up. And this is a relatively fast loop to regenerate the nutrients again. And we can build, for example, we can build this detritus pool into a model relatively easier. So what we do in our models is that we have this mortality term here. Phytoplankton sort of dies, and the amount of, or at the same rate at which it's died, it's put into a detritus pool, and from this detritus pool, nutrients are slowly remineralized, and I've put, first of all, the mineralization is a loss of detritus, and it's a source of nutrients. So this is how you balance this system. And if you do the same sort of simple nutrient phytoplankton detritus model again, I have set the mineralization term, so the regeneration term, to 0 here, just to show you what happens in the model. Of course, nothing, the phytoplankton growth doesn't change, but now we have a detritus component, which starts here with a concentration of 0, and then as your phytoplankton dies, you have an increase in detritus. And if you include mineralization, then you have a growth also during summer, and this is basically fueled by the nutrients that are regenerated through detritus. So this is a very primitive way how you can model nutrient regeneration, but it sort of is able to work, and it's also for models, it's a nice way, because now, especially if you go back to this one, because now the nutrients that were in your initial nutrient pool here, they don't simply disappear from the model, but they are captured within the detritus pool. So you finally maintain a mass balance, and this is very, very important actually for biogeochemical models, that you maintain trace of all the nutrients that you sort of have in your model system. So, but how does that actually work, for example, in BALTSEM? This regeneration loop for detritus, through detritus that I have shown you in this symbolic sort of easy simulation, is not very important in BALTSEM. Most of the pelagic nutrient regeneration in BALTSEM works through the heterotrophic compartment, and that is sort of simulated like a model zooplankton, or it's something between a microbial loop and a zooplankton thing. So how it works, basically the heterotrophs graze on phytoplankton, and they also consume detritus. And they assimilate part of the food, and about 1/3 goes back to detritus, which is unassimilated, undigestible, and so on. And of this assimilated part of it is used for growth, and the rest is excreted as ammonium and phosphate to the water column. And we have sort of the fraction that is excreted is very temperature dependent. So in warm water we try to simulate something like a microbial loop. So we assume that this growth is very inefficient and a lot of nutrients are actually regenerated. And in relatively cold water, they function more like zooplankton. And we assume that the zooplankton compartment strictly maintains its nitrogen-phosphorus ratio. So in our model, phytoplankton has a fixed NP ratio, so it grows according to Redfield ratios. But the detritus pool, those are actually two variables. This is a detritus nitrogen and a detritus phosphorus. That means that the NP ratios in the food to the heterotrophs is different from the phytoplankton and can also vary in time. And how this heterotroph component works, it basically compares the food composition to its own body composition, and then it has a maximum fraction that it can use, and basically excretes all surplus nutrients.
So you have a higher sort of trophic efficiency or something like that in cold water?
Yeah, exactly. Yeah, and also in deep layers. And during summer, this heterotroph, and especially about the thermocline, it just functions as a recycling machine. It doesn't really function like a zooplankton. Okay, so as I told you, this there's also a nutrient regeneration loop through detritus, but this is not the main point of detritus in the model. The main point of detritus in the model is to get material down into the sediment. And the sediment compartment is actually what keeps the long-term storage of nutrients and sort of also the memory of past eutrophication in the model. And this is a plot this is actually the Gotland basin. And this is a plot how the nitrogen and the phosphorus pool in the sediment developed starting from 1850 to 2000. You can see for both pools that basically have an increase. And the sediment pools of organic nitrogen and of organic phosphorus, they are slowly remineralized. And what happens with the remineralized nitrogen and phosphorus in the sediment, that depends on the oxygen concentration in the overlaying water. So basically what you have pools of sediment nitrogen and sediment phosphorus. And what you do, depending actually, on temperature, that was not what I wanted. Depending on temperature, you mineralize a fraction of those pools, and not depending on temperature now, but basically another fraction of those pools you bury permanently. This is basically, you say those are not degradable or not mineralizable within the time frames that you're looking at, so they disappear from the model. It's one of the nutrient sinks that the model has. So the fraction that you mineralize, what happens with that? So this fraction is for nitrogen split into 3 pathways. Part leaves the sediment as ammonium, part is nitrite, and part is denitrified. And the split between those 3 pathways depends on the oxygen concentration. And the same for phosphorus. Part leaves the sediment to the water column, and part goes basically back into the sediment phosphorus pool, so it's stored in the sediment. And the splits, they look approximately like that, so they are pretty distinct functions of oxygen concentration. So here I've plotted the ratio of each pathway to the nitrogen mineralization rate. If you are in an anoxic sediment, basically all the nitrogen that is mineralized from the sediment leaves the sediment as ammonium. And the more oxic you become, the more leaves the sediment as nitrate. And basically the rest is denitrified, so you have the highest denitrification rate if you have oxygen concentration a little bit above 0, and then the denitrification rate declines. And also in fairly oxic conditions, BALTSEM has some kind of baseline denitrification rate in the sediments. And the same for phosphorus. There we have a little bit different. There, basically the release of phosphate is also a function of oxygen conditions. So if you're in very anoxic sediments, you basically release all your mineralized phosphorus to the water column, and if you're in oxic conditions, you release only a small fraction of it, and the rest you store. And if you are in very anoxic conditions, you also release a part of the phosphorus that you have previously stored. So your phosphorus release is higher than your instantaneous mineralization rate. And what we have also built into the BALTSEM sediments is that the phosphorus dynamics depends on salinity. And this is sort of a try to mimic that phosphorus has a different mobility in freshwater and in saline systems. So if you remember, our model has to function in the Botany Bay, and it also has to function somewhere in the northern Kattegat, where you have very saline condition, and in the Botany Bay you are almost in freshwater. And basically, the way that Baltic sediments function is that in very low salinity conditions, the phosphorus release is much less. So phosphorus is less mobile than under saline conditions, and this is a little bit, we have tried to shift those curves according to salinity to mimic this effect. And it has actually created some surprises in climate simulations when some of the subbasins have tumbled over this salinity threshold and suddenly have started storing phosphorus instead of releasing phosphorus. So, and this is one of the things that is, for example, I think pretty much uncertain how that should actually function and how this transition between freshwater and between fresh and more saline condition actually works, because it depends somehow on iron dynamics, and this is something that is not modeled explicitly. So, other biogeochemical stuff. Okay, there is of course one process, it's nitrification. That's maybe not so interesting. It's just a function of oxygen and also of the ammonium, of the concentration of ammonium. And the other process that is pelagic denitrification. If you have nitrate in an anoxic water column, you're going to have part of it's going to be denitrified, and the denitrification rate is simply a function then of nitrate and oxygen concentration. So the more nitrate you have, the faster the rate becomes, and if you are in oxic part of the water column, there's of course no water column, there is no denitrification in the water column. So it's fairly simple. Okay, so how do you then link the biogeochemistry and the physics? How do you link? So basically, and this is the same for our sort of layer basin sort of box model type, and this is also a little bit how the grid points in Marcus model perhaps work. Basically what you do, you model a mass balance. So if you think about your model layers as little boxes, you keep track of the amount of water that flows into the box. And since it's a chain of boxes, you also know the concentration of the substances you're interested in in your input. And from the physical part of your model, you know the amount of water that flows out of the box. And what you assume is that your box is well mixed, so the concentration of the output is the same as the concentration that you have in the box. So this would be a simple mass balance for a conservative substance. Basically, you do what goes in minus what goes out, gives you the rate of change of your pool in the box. So the volume times the concentration is the amount that you have in the box. And those type of models have been applied, for example, also to simple systems. A famous example is Knudsen's salt budgets for the Baltics, basically, for the Baltic Sea and basically also for fjord, where you have a system where you basically make a salt budget for a system that has a freshwater input and saline inflow in the bottom layer, and you do a mass balance for water, the outgoing. The outflow is the sum of the freshwater inflow and the sum of your bottom water inflow, and you do a mass balance basically for salt, and then you can estimate those. If you know the salinity in the inflow, you can estimate the water fluxes that you're missing here. Basically the inflow of salt water and the total outgoing flow. But what we are more interested in is what does it look like if you have a system that has some biogeochemical reaction that go on into your box, and then basically you expand your system. You say the rate of change within the box is now what flows into the box minus what flows out of the box plus a reaction term. And this is basically what is described by all our biogeochemical reactions that we have discussed. And for example, I'll skip those now. For example, for BALTSEM, you write this series of equations for 14 state variables, and for all the layers and boxes that you have in the system. And the 14 state variables that use is salt, then there's a mass balance basically for temperature or heat. Oxygen, ammonium, nitrate, detritus nitrogen, the sediment nitrogen pool, phosphate, detritus phosphorus, the sediment phosphorus pool, and you do it for your biological components, so for cyanobacteria, diatoms, flagellae, and zooplankton. And so basically what you have is a systems of mass balance equation for those 14 components. And then this you solve in time. Okay. What do you do with your model? How do you validate something like that? I think Markus has actually shown a lot of the basic principles already. So one thing that you can do is, does your model resolve sort of the seasonal dynamics of nutrients and phytoplankton in your system? And does it resolve some of the gradients that you know that exist in the Baltic? And this is a plot, for example, of the dynamics of the phytoplankton groups and also zooplankton in the central Kattegat, in the Gotland Sea, and in the Bothnian Bay. And what is plotted here is basically total phytoplankton, and this is a plot that is separated by species. And for example, you can see that you resolve a spring bloom, and then you resolve some reduced summer growth. And for the Gotland Sea, you resolve spring bloom, you resolve some growth in summer, which are cyanobacterial blooms here actually. And for the Bothnian Bay, you can see that you pretty much reduce to one bloom, which is in the middle of summer, so it makes sense. And you can also see that, for example, your spring bloom shifts, basically appears somewhere in March in the Kattegat, it shifts to April in the Gotland Sea, and then something to summer in the Bothnian Bay. You can see that the magnitude of blooms is sort of right. And sort of you can see that you have a succession from diatoms to the summer species, and you can also see that you have the zooplankton bloom, the zooplankton peak somewhere in the middle of the year. So that sort of looks alright. So this is what you can do. What you can also do, you can sort of eyeball what you have for basins with very high vertical gradients and basins that you think are important for the biogeochemistry, which is of course then the Gotland Sea in the case of the Baltic, and then you can prepare plots, long-term plots. This is from 70 to 2006. Those are plots of observations of oxygen, nitrate and phosphate, and those are plots of your simulations, and you can sort of just visually compare whether you have the right timescales, the right gradients, and you can see some nice agreements. For example, in oxygen, you capture the major stagnation period. You also capture that you sort of have, when there was an inflow, you have basically oxidation of all your ammonium to nitrate. So you see that both in simulations and observations. You can also see that you have some difficulties. For example, this was this long Baltic stagnation period where you have a high accumulation of phosphate. It's much higher in the observations than it was in the model. So this is sort of qualitative. But what we also try to look at is that some kind of long-term trends. What we have used is winter nutrients and summer phytoplankton. The winter nutrients, for that reason, that we think that they sort of reflect the long-term dynamics of the nutrient pools in the Baltic. And this is just plots for nitrate and phosphate in the Gotland Sea, and just as an example in the Gulf of Riga. So those are the observation, and you can see that some part of the trends are captured. For example, for the Gulf of Riga, you get this nice increase in winter phosphate. You can see that, and for nitrate, I think the Gotland Sea, that's also sort of nice. You get this increase and then a decrease, and then you see some disagreements. And the same is with phytoplankton, basically. And there I just put some plots that shows the problem. It's very easy to get nutrient data, and then when you move up sort of to chlorophyll and to zooplankton, it gets more and more difficult. The data is harder to access, and it needs a lot of work in pre-treatment. So it's actually no fun to try to do this kind of validation stuff.
So you have your biomass as nitrogen, don't you?
Yeah.
And so you use the fixed ratio nitrogen to chlorophyll?
Yeah.
Okay.
Yeah, a fixed and very rough ratio.
What's the number?
I think we used something pretty low for chlorophyll to carbon. It was more on the low end. That's what I remember. But
so you use also a nitrogen carbon ratio, and through that go to chlorophyll.
Yeah, if you look at published data, you will find it basically as a chlorophyll carbon ratio, and the model doesn't explicitly have carbon.
Yeah.
But you have some basic assumptions that it's red-filled carbon for the phytoplankton, and that shows up in some kind of the oxygen equation. So that's sort of consistent.
But you have a parameter which is nitrogen chlorophyll, and you put some value for that.
Yeah. But only in processing the output.
Okay.
It actually never shows up in sort of state variables. It's just the derived parameter. What we also try to do is something similar to what Marcus showed, to sort of summarize the model bias. And this is the same thing, blue and green where we think the model performs well, and red where we think the model performs bad. So if you look, what did we have? The Bosnian Sea and silica, right? And phosphate. So even in the new BALTSEM, we don't do so no. Bosnian Sea is this one. So it became a little bit better, but it's still not super good. This is a quick and dirty way just to look, and especially if you calibrate models, you want to have one number that shows you, oh, this model parameterization is better than the last one. You don't want to have it so that you don't have to go in each of the nitty-gritty details. But this is really no fun because there's a lot of work in it to do this model data comparison. It's not so straightforward. What you also try to do is that you compare some fluxes to published estimates. So one candidate's primary production, nitrogen fixation. You can find in the literature estimates of sedimentation rates, denitrification, and also some sediment water fluxes of nitrogen and phosphorus. You can find some estimates for permanent burial rates in the sediment. But it's always, we have used those as a check whether our model does something reasonable. But the problem is, how do you scale from point type of measurements to basin scale fluxes in a model? So that's not so straightforward. Okay, after all, we also have some modeling fun. And I think the greatest modeling fun that we had is actually was this long-term hindcast. This is part of the simulation that Marcus has also shown from 1850 to present, where you can see the long-term response of the Baltic. And the nice thing is that you can really see if you have this Baltic in 1850, and this is oxygen here, and all the blue values, I think they are anoxic. So you can see that you start with a very oxic Baltic, and you become anoxic basically, starting from the 60s, 70s. And you also see a change in the model in primary production and in phytoplankton, and then also in phytoplankton concentrations, of course. So you start with a low productive system and you get much more productive here in the end, and you can also see that in zooplankton, and of course also in the nutrient pools. And for other modeling fun, we have tried to, you can also try to do fun simulations. What would happen if you simulate the long-term dynamics of eutrophication till present, and then you reduce the loads again to the loads as they were in 1850. So would you get your the state of the Baltic back as it was, or would it be something different? And how long would it actually take? And I've tried to characterize the state of the Baltic. Sorry, you can't see the grayscale really here. With some plots. The variables that you see, the red stuff here are oxygen, and you can't distinguish between red and brown. This is bad, because brown is salinity and the red stuff. This is actually red, so this is oxygen, and this is temperature, this is salinity, and the green stuff, those are phytoplankton variables, and there are some orange bars, this is zooplankton. And you can move through this system and see how it changes, and variables that are plotted close to each other are variables that are relatively highly correlated. So those are very relatively similar in their response. And what you can see, this is the very oxic system to start with. You can see this is basically 2020 to 1940. You can see there that you have an increase first in nitrogen pool and bottom water nitrogen actually. You have an increase in phytoplankton growth and an increase in zooplankton growth, but you're still fairly oxic.
Is the length of
the length of the bar
just
is it's min-max. So it's for the entire length of the simulation from 1850 to 2098. If the bar disappears, it's at its minimum, and if it's out here, it's at its maximum. Oops.
Should we have a break and continue after the break?
Yeah, I'm almost done.
Okay.
It's just a quick walk. And if you move on, this is the system in the 60s. Here, basically, your oxygen starts to disappear. You have the first signs of anoxia and hypoxia. You have nutrient accumulation in the surface also, and you start to accumulate phosphorus, and you get more productive. And this is the system, as the model says, we have it right now, full of nutrients, very productive. And these bars here are cyanobacteria, so also with high cyanobacteria blooms. So now if we set the nutrient concentrations back to the levels in 1850, and we start to implement that stepwise between 2007 and then I use some 20 years as a transition period. Actually, the first effects in the model you see already in the time period 2015 to 2030. You get less productive. But you still have a lot of nitrogen fixation, and you still have a lot of phosphorus in the water column. And if you simulate, and you still are fairly anoxic, and if you simulate onwards, what happens is, first of all, you get more oxic again. You are still very productive, and you still have quite high phosphorus concentrations in the water column, but also your bottom water nitrogen pools start to accumulate again. And at the end of my simulation, oops, was it already at the end? Yeah, it was almost the end. So at the end of my simulation, it doesn't look very much different. You basically still become a bit more oxic and a bit less productive. And the time period to 2098 is not quite enough to return to the initial state, but actually almost. So in our model, the Baltic is very reversible, but the return pathway is not quite the same as the pathway you started out from, because the phosphorus pools remain to be fairly high. So this is modeling fun. And then what the model is actually also used for is serious business. And because you can run a lot of scenarios, it's now used for doing for designing the Baltic Sea action plan, for doing country-wise nutrient reduction scheme. So BOSE has been running a lot of scenarios to calculate which countries should reduce the load to which Baltic sub-basin by what, and I'm not sure how many simulations in total he has run, but it was a huge work. So this is a serious business in modeling, and also it has a serious consequences, because it's really a requirement to the countries in the Baltic to reduce nutrient loads according to what we have calculated in our model. Okay, so that's it.